and go ahead and introduce our panelists and get started with our medical panel. So thanks everyone for joining us. Thanks everybody for a good day so far. A few little glitches, but you know, this is how these things go. So we have a great panel. Bill Gahl is going to be acting as our moderator. I'll be helping him identify questions, and I've been gathering them from the other sessions, and we'll be trying to address everything. And please feel free as we go along to add questions into the chat, and I will make sure that they are answered to the best of our ability. Dr. Gahl will be our moderator today. Our other panelists include Dr. Craig Langman, Dr. Paul Goodyer, Dr. Jess Thoene, Dr. Rachel Bishop, Dr. Paul Grimm, Dr. Rick Kaskel, and Dr. Doris Trauner, and Dr. Larry Greenbaum. So with that, I'm going to leave it to Bill to get started, and we'll start to answer some of your questions.
Well, Christy, I think you were going to feed some questions to me, but maybe we should just open this up to all of the non-panelists to see if they have any questions.
Well, I can get started with a few that I have. I didn't know if you wanted to say a few words. And I'll start with one. And like I said, please, everyone in the meeting, feel free to enter questions in the chat. So I'll start with this. We had a question in one of our Parents of Adult panel to ask about what research is going on in regards to the muscle wasting. What, if any, research is going on to the research or to the issue of muscle wasting in cystinosis?
Well, I think maybe Doris, can you fill us in on an update on that? Because I would say that I don't know of any that's being done at the NIH.
I actually don't know of any. There is research going on in Boston. I'm blocking on the name now. But I think it's right now collecting data on natural history more than anything, and on kind of baseline evidence. Oh, Mass General. Okay, thank you. Yeah, but I don't know of any treatment studies going on.
I do know that people like Katharina Hohenfellner is doing studies on the influence of exercise on the muscle of patients, and finds that exercise may be as important or even more important with respect to the myopathy than cysteamine itself. And it makes a little bit of sense. That is to say that the muscle can still hypertrophy as long as it's still there. So I think that's one thing that's going on. But otherwise,
Bill, isn't she doing the rotating plates?
Yes, she is. Yes.
Yeah.
That's right. So in other words, it's a specific type of exercise that may be more beneficial to the muscle than, let's say, bench press.
Right. And as an added note, Dr. Hohenfellner will be presenting her findings to date in that study. It's partially in part funded by the CRN, and that'll be taking place tomorrow. So for more information on her work, you can tune in tomorrow.
I thought that Larry Greenberg was also doing something similar, just the impact of exercise.
Larry's here. Go ahead.
Yes. We actually we did a study thanks to the CRN. We did a study, mostly a combination of our own patients, but a large percentage of patients were patients that we entered into the study at the last two in-person CRN meetings. We just did grip strength testing as a way of assessing muscle strength. We compared this to national data. We corrected for height and age, which is actually fairly complicated because of the tremendous effect height has on muscle strength, and age as well. And we also compared this to similar data that we analyzed through the C-KID study, which is a study of children with kidney disease. And unfortunately, what we found, and again, this was cystinosis patients from age 8 to in their 50s, so it's a really wide range, was that cystinosis patients, as one would expect, have significant deficits in muscle strength. What was interesting was that the males had much larger deficits than the females. And as I said earlier on another panel, that doesn't mean that the males were not as strong as their females of the same age and height and so forth, but compared to other males, healthy males, they had a larger deficit. And one hypothesis on this was that potentially the effect of testosterone and hormones like that may be having an effect, because we know that testosterone influences muscle strength. So again, it was just observational. We have no solutions, but it emphasized, I think, as has been shown before, the magnitude of the problem.
Yeah.
And I want to thank the CRN for allowing us to do that study, and all of you as patients and family members who enabled us to do this study.
Yeah, no, that's our pleasure, Larry. And that's one of the things that we don't get to do, not being in person, is participate in studies. But hopefully in 2 more years we will. So Dr. Gahl, I see another question in the chat. Perhaps you'd like to address this to someone on the panel. It says, how do we improve the quality of life for cystinosis kids, especially those less than 10 years old, as they have multiple medical conditions and they are not active as other normal kids?
Well, people need to encourage activity in any way that they can. Maybe some of the members of the panel have some particular methodologies to improve the activity of kids with cystinosis.
I'll just mention that I think that the use of physical therapy is really valid. I don't know if that's exactly the theme of the question, but most of the academic medical centers where the patients are seen, I'm sure, have excellent physical therapy departments that can tailor looking at the child, do an assessment, and then give some great examples of how to get more activity from the patients, with the patients.
And I think that children with cystinosis may sometimes be in too protective an environment. They actually have lives to live. And I guess maybe all parents are a little bit protective. Some are more helicopterish than others. But for cystinosis, they have to grow too. And so maybe we can encourage our families to allow the kids to have a more normal life and not worry about running around outside and things of that sort, or even engaging in some of the mild sports, as long as it's not, roughly speaking, hockey.
Or tackle football.
Nobody actually
should play. I agree with you. Yes.
I know. From a pediatrician standpoint, tackle football is not good for, especially when you get to be 50 years old and have no knees.
Bill, maybe you could comment on the risks involved in softball.
Yeah. Well, I've been knocked unconscious 3 times in softball, and I broke my jaw playing softball. So it's risky, but only if you play the way I do. So nothing to worry about there.
Bill, I would also say, optimizing things as much as possible. We talked in the last session about the fact that a lot of children with cystinosis have, quote, flat feet and can benefit by orthotics. And so making sure that you do all you can. If they have orthopedic problems, making sure that those are corrected. And finding activities that fit. I had one patient who was a huge baseball player, loved to play baseball, but at some point, and I had the same experience, not being very tall, it became more difficult to compete. And he became an avid golfer, and that was the sport for him, and it worked out well. And I played soccer where you don't have to be that tall. So it really, find things that they can do and encourage them to do it, optimize their physical health. And I think what Bill said is really critical is, to try to treat them as normally as possible, and not be overly protective so that they can participate in things. I know that can be very, very difficult.
Rachel, you were going to say something?
I was just going to add the psychology kind of angle on this, two points. And one is, I think as we evolve also over these years of being together as a cystinosis care community, integrating these questions into our discussion. So you're here, you're visiting us, I'm the ophthalmologist. If I'm asking, what are you doing for sports? That's triggering that. And I'll say, I haven't asked that until it started coming up, Doris, from your presentations and others over the years. And so I think it's on us more from the physician side to be integrating it into the dialogue we are having. And for you all on the family and patient side, for those I'm seeing here in the chat, great examples of all the sports you've been playing. We have these all the networks that patients are in, families are in, kind of highlighting your own sports experiences to be role models for those who are younger and wanting to enter sports and feel comfortable taking those risks. I think just the awareness on both ends, the patient and family side and the provider side, will help that be part of the dialogue.
Yeah, I see in the chat that the sports, soccer, swimming, cheerleading, basketball, all I would say reasonable, and orthotics and physical therapy, the things that are very helpful for our cystinosis youth. Paul, you have a hand up.
Yes, I really wanted to emphasize that oftentimes parents are protective and they just don't want their child to go to any harm or any problems. And also doctors who don't know cystinosis well might be too protective. Oh my God, there's a little bit of rickets, don't do any sports. Oh my God, you have a kidney transplant, you shouldn't do any sports. That's wrong. And I think we as specialists need to really be advocating for patients. Yes, if you have a kidney transplant, you can play in the NBA. Yes, as a kidney transplant patient, you can do competitive soccer and swimming and basketball. And so I think that we just need to be aware that doctors who don't know it well might be a little bit too careful and protective, and we need to balance that. The second point, I see a lot of loneliness in adults with cystinosis. And if you can take the opportunity to get them to participate in team sports at a younger age, that might help them with their socialization, because a lot of these kids have lost a lot of years in school or are homeschooled. And again, those are opportunities for socialization which they lose. So encouraging socialization, encouraging team sports can help in other domains rather than simply muscle strength. And the last point I wanted to make was upper body sports. Like many people might run or jog, which is great, and even play soccer, which is great, but it's maybe not so good for upper body. So just don't forget the upper body, because that's where we see troubles with the hands, so like rock climbing or things where you're doing stuff with your upper body, or even playing guitar and stuff like that. Don't forget those as well.
I think this is all a great conversation, and there are a lot of great points or suggestions in the chat. Bill, we have a couple of research-based questions. I don't know if we can move to that. First one, someone asked about Paul Goodyer's nonsense mutation study. So I don't know, Paul, if you're able to give an update on that.
Sure. So as you know, we were using a read-through drug related to gentamicin to try to activate expression of the C2S protein in people who had a special type of mutation. And so we started that study, and we got through the first 3 people at a relatively low dose. And I think there were 2 things we discovered. One was that the threshold for efficacy was about 1 milligram per kilo per day per dose. And this, just to put that in ballpark, we have a frame of reference. We use about 7.5 milligrams per kilo as a single dose of gentamicin. So it's well below that. But when we went below 1 milligram per kilo, there was no effect. And so I think we identified the threshold where it was starting to be effective. But then the pandemic came, and it's all been on ice ever since that time. I think that our plan is to go back at that when we are allowed to, when we can't yet. But we're going to have to revise the protocol, because we found that there was, when we started, we tried to wash out the effect of cysteamine, and we thought that a week would be fine. But it turned out that in the ensuing weeks, the baseline white blood cell cysteine just kept rising and rising and rising. And so it was very difficult to run the study the way we did. So when we get around to it, what we're going to do is to change the protocol and keep everybody on cysteamine, and then make sure that the white cell cysteine can drop when we introduce the additional read-through drug. So it'll have to be redesigned and run a little bit differently to take into account this problem of the rising baseline. And I think it has to be done though, because we can't take people off cysteamine for a month or month and a half before we start the study. There. So I think we have a plan, but no clarity about when we can actually put it into action.
Okay,
So-
thank you for that. Go ahead, sorry.
No, that's it. I mean, that's as far as we got.
I appreciate that. I know that was on the fly, but I suspected you might get questions about what was going on. So other research questions. So, and Dr. Gahl, you can maybe frame this however you'd like, but we have a lot of questions about gene therapy and what's going on in Dr. Cherki's study and the AbroBio study to come. Lots of questions about the panel's opinion on if it looks promising as a cure, and if it would be more beneficial and available for younger patients as it moves along. So I don't know how the panel would like to address the general topic of gene therapy, but I think there's a lot of interest.
I think I might ask Paul a little bit, but I don't know an awful lot about this. I know that there were 6 patients who could be enrolled in this. 3 have been enrolled so far. They're all adults, except for one who's not quite an adult, I think. But in any event,
He's only 18. Yeah.
18, yeah, okay. And I think there are plans that we'll probably hear about tomorrow from AbroBio to do kids who are 18 months and older or something of that sort. But I would say Craig or Paul Grimm or someone else, if you have more knowledge, I'd love to hear it.
Well, I'm happy to talk from what I do know and can share publicly. I am a consultant for them, so I just want to get that out of the for fair balance. The first patient was a patient of ours who's done exceptionally well. He's on the website, kid named Jordan. He lets his name be used, and he is actually off all of his standard medications and has had marked crystal reduction in multiple organs when biopsied. The second patient was another patient of mine who has actually had 2 kidney transplants, and except for a little graft versus host disease with the very first stem cell transplant, has done remarkably well, and his Fanconi syndrome has disappeared, and he too is off all medications at this point, and has this kidney is the second kidney transplant is perfectly normal. He has no pathologic protein, no hypertension, and is doing exceptionally well. So those are the 2 patients I've been involved with that I know about. There have been, I think, 2 more? Rick, do you know of Rick or Paul? 2 more patients?
Yeah,
I've heard that, but I don't know.
there are. I'm a co-investigator in the study.
Yeah.
There have been 2 more patients.
Yeah.
So
Are they-
Craig, the Fanconi syndrome disappeared in patients who had 2 transplants already? Why would they have Fanconi syndrome?
He still had loss of tremendous amount of phosphate with his first transplant, and obviously the new kidney wouldn't have Fanconi syndrome, but he still had lots of mineral loss, which is unexplained to me, to be honest with you.
Yeah, okay, well, that's why I asked, because it seemed
Yeah.
the only reason that would occur ordinarily is if the native, the host kidney still had some function in blood flow.
Right. I also think there might have been some toxicity from some of the immunosuppressive drugs that can do that too, so.
Okay. So Paul Grimm, do you know anything more about this, or does anyone else
No.
know more about this?
You know, I think Doris is the most up to date because she's one of the co-investigators and at San Diego. So I would go with her as the gold standard in this group.
Yeah, okay. And Doris, I guess you already mentioned that you don't have a lot of information that you can give us
Right.
on the individuals.
Right.
Okay.
But I think that they are going to be dropping the age down to 12 or something for the next cohort. Is that something like that?
You know, oh, go ahead.
No, I think they're talking about that. I don't know if that has been approved by the FDA yet. I just don't know. We'll find
As
out.
far as I know that,
Yeah.
as far as I know, that hasn't been approved yet.
Right.
Yeah, and we will have a presentation from EveroBio directly, which will be a sort of first. Previous updates have been from Dr. Cherki, and so we're all interested in seeing their presentation tomorrow. So hopefully that'll give us more information. And I know they can only share so much, but it is good to get the panel's opinions. As treating physicians, as investigators on the trial, you can only give so much. But
I'm going to go out on the limb, and
Okay.
not because I have any relationship with them, but I think that actually a stem cell transplant will become the standard of care in the next few years for this disease.
Yeah, I think a stem cell transplant has some possibilities. I think that if it's a hematopoietic stem cell transplant, then what we're asking those hematopoietic stem cells to do is either communicate directly with other cells to provide the cystinosin to the other cells, or else give rise to parenchymal cells that are not of the hematopoietic lineage. And both of those things, I think, are possibilities, at least for some of the parenchymal cells involved. But I have a concern that in order to have clinical efficacy, those two mechanisms and processes have to occur in many, many, many cells. And I'm not sure that that's going to obtain.
So Fernanda Copeland, who's the patient advocacy representative for EveroBio, is here. I've asked her, and she has made it, she's let me know that she is available for questions. I've asked for our technical support from CVent to unmute Fernanda. Fernanda, can you unmute yourself? She may not be able to. Fernanda, you can interrupt us once you're able to, and maybe we can get her to answer some of these questions today as much as she can. She's not able to unmute yet, but she's hopefully our folks at CVent will unmute her and she'll be able to join.
She may not be available. I got a direct message from her. I'll be available for questions tomorrow, so maybe my.
She's sending me one that she is available now, so we'll see.
Oh, good.
Okay,
We'll
so
see.
just feel free to interrupt us, Fernanda.
Yeah,
And maybe-
once we get that worked out, feel free to. Anybody else have an opinion on this? Otherwise, I'll move to another question in the interest of time. Okay, we'll wait till we get Fernanda for any more follow-up. So, Bill, we do have a couple of different topics. I'll start with the one that was added first, and you can direct this to whomever you think most appropriate. There's a question in the chat. Is there a consensus on using supplemental testosterone in male patients for growth and musculoskeletal health? If so, when's the best time to?
Someone like to jump in?
Yeah, actually, I'll just jump in, and I have a patient on supplemental testosterone with the help of our endocrinologist, but I wouldn't do it until the teenage years. So I don't think in very young kids that would be something I would want to do.
Yes, and I think the other prudent approach is to measure testosterone and replace testosterone in those people where the levels are too low, rather than give it to everybody or
Yeah, of course.
just because of their loss of strength.
Yeah, I guess that I would summarize by saying that there is no actual consensus for this because every individual is an individual with different testosterone levels, different ages of puberty, different levels of muscle involvement, and different severity of cystinosis. So that would be my answer.
Okay, thank you for that. So let's move to a different topic. And since we have Dr. Rachel Bishop here as our ophthalmology expert, we've had some questions come in, some specific ones and others. And maybe you can sort of give us some of your viewpoint generally on the use of eye drops, since we have you here and we haven't had a really specific session on it, the two different formulations, what your opinions are. And then we've had specific questions from folks about, for example, there's one, after 8 months with Cystadrops therapy, my son's eyes started showing crystal signs in his iris in both eyes. We put drops in constantly, never miss a dose. So I wonder why. And I think that type of question is probably prevalent, in lots of folks and parents and individuals. And maybe you can sort of address the topic of eye health generally.
Sure. Thank you. And I think conceptually, what we're doing with eye drops, and just to make the distinction, why are the eyes separated and treated separately? The oral medicines get to almost every cell in the body. That is every cell that has a blood supply. And the ocular surface, the cornea, the clear part of your eye that you look through, does not have a blood supply. So we have to treat it separately. So oral medicines don't reach the cornea, which is why eye drops need to be used to leach out the crystals. So that's the quick big picture. And the effectiveness of different formulations, whether they're more liquid, more viscous, thicker, has to do with the contact time on the surface being treated. So if you have gel and it's sitting on the corneal surface and it sticks around longer, there's more opportunity for the cysteamine to work and dissolve the crystals. A liquid form sticks around only a few minutes and therefore has a shorter opportunity to work. So they're really, the mechanism is the same. It just has to do with contact time between the Cystaran and the Cystadrops. I think the naming is a little unfortunate in that it causes confusions. The Cystadrops are the thicker one that are the more gel-like, and the cysteamine is the more liquid drop form, which is the formulation that was studied at NIH in the trials and approved initially. Regardless, some patients seem to do better with one versus the other. Some tend to tolerate one more than the other. And like many areas of medicine, we like to have more choices so that we can tailor therapy to what works for that particular person. So I have no opinion to say this one's superior to this. It has to do with what's tolerated. And I can say I've seen examples where people tolerate Cystaran, do not tolerate Cystadrops, and a little bit the other way around as well. I'll also say, full disclosure, my experience with Cystadrops is quite limited, and it's in the clinics I've seen in Germany more than anything. Now that it's in the United States, our experience should grow. So that's sort of the overall picture. Use what works best for you. Of course, you also have to use what's available. And the second kind of word of guidance is the frequency that's required, as Bill said, varies by person. A few tips to the question of why one might work for one person and not another. If you apply a drop of any form and you blink very strongly right after the drop goes in the eye, you're blinking most of it off the ocular surface, like a squeegee on a glass surface. So what you want to do is put the drop in and allow it a little contact time, blink gently. Just tip number one. To the question of, and the other thing that we've been saying a long time, but for new family members hearing this, the FDA approves a certain regimen, and in the eye drop for Cystaran, for the liquid portion, it was approved every hour while awake. And we have heard the messaging, and I'm a strong believer in this message. This should be tailored to the lifestyle of the individual. These administrations can be clustered more closely. If you're using Cystaran, you can do 4 doses in the morning, spaced half an hour. if your eyes seem to tolerate it, if they don't become red and irritated. You're not hurting your eyes by dosing more frequently than the interval of time that's been approved by the FDA. So you're sending your kid off to school. You can dose more frequently in the morning. They go to school, they come home. You can dose more frequently in the afternoon and evening hours, provided the ophthalmologist is confirming that you cleared the crystals and they're remaining cleared on that routine. For other kids, they will need administrations during the school day because for whatever reason, that routine isn't enough. So 6 to 12 times a day on the more liquid formula and 4 times a day on the Cysta Drops gel formula seems to work well. So there's that. That's addressing the frequency and the spacing. And if you happen to find I'm doing them more often and now my eyes are a little irritated because the preservative is getting in so close in time that it's irritating me, then you're somebody who has to spread them out more. Again, all of this is one of the reasons why you have to continue the eye exams each year, or more often at the beginning, or more often if you're having more problems, so that you can get to a routine that works like every other body part that's being monitored and cared for. Addressing specifically the question of the Suhi here, who the crystals are being noticed in the iris. So the iris has a very good blood supply. It would be speculation for me to say how much of the iris crystals are controlled by oral medicines versus topical. Topical also penetrates the eye, but without knowing the specifics of this situation of this crystal formation in the iris, it's hard for me to respond. Functionally, those crystals don't impair iris function, but what I would use as the monitoring metric would be, what is the cornea looking like, and also what is the conjunctiva looking like, the white of the eyes. I hope that's helpful.
Yeah, let me mention 2 little points. One is that the active ingredient is the same in all of these. It's always cysteamine. And secondly, that concentration of cysteamine is huge relative to the concentration that's achieved in the blood by giving oral cysteamine. The highest concentration of oral cysteamine in blood in a patient with cystinosis receiving cysteamine is about 50 micromolar. And the concentration of 0.5% cysteamine is 50 millimolar. You can do the calculation. So that's 1,000 times the concentration.
It's an order of magnitude higher, yeah.
No, it's 3 orders of magnitude higher.
Yeah.
So, I mean, that's just huge. And that's why you can actually dissolve crystals. And remember, this is stuff being put right onto the cells of the cornea, as opposed to blood levels that are 1,000 times less and have to get to the cells that are nearby the blood. Okay, so those are my couple of points.
And Bill, can I add just one more sentence? And again, just to new CRN members, and so great that there are new members here, which is the final punchline is these work. They totally work. We have patients who came in on the Bill Gahl grading scale, which goes 0 to 3. That was the grandfather of all these studies now so long ago. Patients came in at a 3, like the cornea is packed full of crystals, and got to 0 or 0.25, sometimes in 12 months, sometimes in 2 years. And some people never got there, but they ended up at, a 1 or something, which is a manageable level. So just realize it works. It works. We have a treatment. It works. And if you're not getting the result, don't give up working with the ophthalmologist to get to that point, because they work.
Yep. Next meeting, I'll be a great grandfather, I suppose. No, not really. So, but Christy, is someone on now that's supposed to be?
Yeah, I was just going to say, so thank you so much for that, Rachel. That was a great summary and addressing of that person's question. So Fernanda Copeland from AbroBio, who's the patient advocacy lead, has generously agreed to be on camera, and they will have a presentation tomorrow, but for Bill and Fernanda, perhaps you two can have a discussion. And Fernanda, I know you can only share certain things, but there were some questions that were thrown out, and what you can let us know what you can address and what you can't.
Sure. I definitely am not camera ready, but thank you for having me. So as a backgrounder, the phase 1 clinical trial is being led by UCSD. So we are not the designers or the owners of the clinical trial. But we are very grateful to be learning from that clinical trial to help us select future endpoints and a pathway forward with the regulatory agency, as well as inclusion and exclusion criteria. So the company right now is in the beginning phases of planning the future clinical trial. It is the goal of UCSD and the investigators, the primary investigators, to finish the phase 1/2 clinical trial. It could be up to 6, but not necessarily 6. We, being the sponsor, are very many arms length away to selecting the patient. So we don't know who is on the queue at all. We have no idea. But the company has announced publicly that we are planning the next clinical trial, and we're hoping to open it next year. My colleague, Dr. J, or he goes by Jay, but his name is John Jeffrey Jones, will be giving an update on the clinical trial tomorrow. You have seen, everybody has seen the latest data. We don't have new data to release tomorrow. What we do have is a little bit information on the planning for the clinical trial. I heard from you that there is a plan, there's an interest to including younger children. That's correct. Several sites, that's correct. More than one country, that's correct. But we don't have the final details yet. The company is engaging with regulatory agencies late this year and early next year. And as I heard on the call, many of you are consultants or advisors for the company. And so we're definitely, and so is Christy and the European CAB. And so we're definitely consulting with the experts before moving forward. And we really appreciate that collaboration.
So I wonder if there are any specific questions now for the AVROBIO person.
I don't have anything else in the chat. If anybody wants to enter something into the chat from our audience, we can see if Fernanda is able to answer. I don't know if any of our panelists have anything, in particular the ones that are not. Go ahead, Jess.
In regard to the general life ability of our patients, something that we don't discuss often, and I know why, but as important as the physical health is the mental health. And I don't know, correct me, anybody on the panel or in the audience who knows, that we've ever had a serious session in the decade on mental health issues. Now, I know it's done for children with chronic diseases, and obviously cystinosis is one, but it might be useful even planning to try to do that in a future year.
Yeah, I think that's a good idea.
Especially this year, we've had.
Here's what I found.
Stress.
Yeah, it's such a
It's a
tough
great
year.
idea.
But let me get back to Fernanda and just ask if you know whether or not the data from the first, I assume 3 patients, are going to be published at some time soon.
That sounds like a great idea. As far as I know, we're not working on anything right now, but I know there's other people from AVROBIO who are also listening in, and sounds like we should do that sooner than later. We are building on, as you know, we have 6 programs that we're working on, and we are building on safety from different lysosomal disorders as well that help build the safety data for lentiviral gene therapy. Thank you for that.
So just, for the families and patients out there, looking at the future, the first step is obviously finishing this initial clinical trial. And then what you're telling us is then there'll be another clinical trial that will be much larger. And that will hopefully be used to get FDA approval for the technology. And once that's done, if the FDA approves it, then this technology would be available really to all patients. Is that a good way of summarizing how everybody will get access to this eventually?
That's very helpful, Dr. Greenbaum. So yes, correct. The phase 1 clinical trial right now, it's a safety clinical trial to try to show safety in this population. And then the next clinical trial is a safety and efficacy clinical trial. And sometimes we add different populations. That's why we are considering different ages. And then following that clinical trial, the goal is to submit an application with the regulatory agency so it can be available to more people. But
So go ahead.
We are still in finishing the phase 1 to trial, and that's the next step.
So with different safety considerations in children, especially younger children compared to adults, is it a plan to have an intermediate clinical trial for in cystinosis individuals 12 and up? I mean, I heard something about that.
We're still planning for the next clinical trial.
Understood. Okay. Thank you. Any other? Go ahead.
There was there's a comment, and this is partly relates to Dr. Thoene's comment about mental health and quality of life, and I'll direct this a little bit to you, Fernanda, as well. First, I don't know if you can comment on the inclusion of any quality of life measures in the trials. And 2, we had a comment from Barb Kulik, who is here and has been very generous with her ability and willingness to talk about her son Jordan and his involvement in the first trial. She's added in the chat that she thinks mental health needs to be added to conferences as well as for stem cell transplants in future sessions, life after cystinosis perhaps. And I think that's a really interesting comment and a different paradigm in how we look at the disease generally. But Fernanda, I don't know if you can comment on anything.
Yeah, no, agree. I completely agree. I'm a dietitian by training, so I understand that the medication is just one part of the disease, but there's the rest of the person, the whole person that needs to be considered. There will be quality of life measures. When we met with the Cystinosis Advisory Board, the CAB that Christy mentioned in the beginning, we did inquire. There were a lot of discussion about quality of life measures. So they will definitely be included. And because gene therapy is a little different than other clinical trials, we're also trying to find what is in a different way, maybe a companion protocol or a different way to measure the quality of life of individuals. It has been done in other communities like the Duchenne muscular community, because there's so much more to quality of life that is often not captured in a questionnaire. So yes, we are considering, we hope we can be creative in different ways to measure that and try to support the whole person. And we welcome ideas from you experts, always.
Thank you, Fernanda.
Thank you.
Thank you for jumping in. I really appreciate it. We'll move on. I see we have 15 minutes left in the session. We'll move on to some other topics, but feel free to go off camera now if you'd like. But we appreciate you joining us, and we'll look forward to your presentation tomorrow. So I do have another question here in the chat, and someone has asked us at the group of you experts, for a physician, what could be the ideal age to cure cystinosis disease and prevent others from developing? This is a question from one of our parents.
In utero. Now, you'd have to diagnose it, and then you'd have to do the stem cell therapy in utero, but the earliest possible would hopefully allow any prevention of damage to the organs as they're forming, because the moment the baby has their kidneys working in the womb and after birth, the damage is starting, right? So as soon as possible, if it's theoretically possible. Yeah, it's probably as soon as possible, right?
That might happen in gametes. I think I've read something of CRISPR technology in animal gametes. So in fact, you could precede conception.
Yeah, we might be getting to the border with ethical issues here. I mean, that's possible.
Think so.
You know,
Yeah, go ahead.
I had a thought a number of years ago, really based on the old microdissections of nephrons from the kidney. So if we posit that the kidney is one of the first organs to be badly injured by cystinosis, it looked as though the atrophy of nephrons didn't occur until the second year of life. And so I think that there probably is a window of opportunity between the time we can diagnose it, let's say 4 or 5 months of age, and because the Fanconi syndrome starts, and the perhaps irreversible damage to some of the nephrons by a couple of years of age. There might be a window in there where we could intervene if you had a successful intervention, that would be the window of opportunity to get in before the most sensitive organ is irreversibly harmed.
Interesting.
Bill, I'm not sure, but for some diseases, some people in Europe are doing stem cell transplants in utero. Is it legal in the United States to do that? I'm not sure.
Only to change their voting status.
You know, I should have. Okay. So I work for the federal government, just that I mentioned. But I think the legality is maybe the issue so much, Craig, as it is to the IRB approval of something of that sort, and the risk and benefit issues involved.
But well, the reason I mention is there's another disease, osteogenesis imperfecta, which is a bone disease, brittle bone disease, and some colleagues in Sweden are actually doing stem cell transplants in utero with great success, actually. But when I raised that issue with our bone, the people that do stem cell transplants at our place, I was told that it may not be legal in the United States to do that. So that's why I don't know enough. I'm just asking the questions.
Well, you know, there's government-sponsored fetal therapy, which has been a political issue.
Yeah, there you go.
And so, but that doesn't really answer the question overall, because there's also non-government sponsored research.
Yeah,
that's performed, but lots of times the non-government research goes hand in hand, or it can't be done, without government support.
Interesting.
There have been a couple of questions, Bill, and let me just throw this in there and then continue with your conversation.
Okay, yeah.
A couple of comments and questions. One asking about amniocentesis. This is a preferred testing method, obviously in families that already know that they have a child with cystinosis. And then a comment from Dr. Nesterova about newborn screening and how that might apply to what we're talking about and the possibilities there.
Of course, amniocentesis occurs later than the possibility of chorionic villus sampling. But I guess if you lump those two together, prenatal diagnosis can be performed on cystinosis. I mean, that we've known that for probably 2-3 decades or so. And the issue of newborn screening, I think, I've said this probably for about 10 to 20 years, that the next major thing that we need to do for the field is to do newborn screening for cystinosis. And it's a very difficult thing to do based upon the current technology, which is mass spec analysis of cystine levels. And the reason is because the amount of cystine that's in the plasma portion of the blood is so much higher than the increment or the increase that's present in the white blood cells of cystinosis. So there's this huge background of cystine that doesn't allow us to measure based upon a blood spot from a heel stick on a filter paper, the difference between a cystinosis and a normal. However, molecular diagnostics can be performed, and actually in Germany has been performed. And Dr. Hohenfellner has done that study along with her colleagues, and they had 200,000 German newborns tested, and one of them had cystinosis, and they had, a huge number of heterozygotes, people who had only one of the abnormal genes. But that could provide the basis for a newborn screening, which would revolutionize the treatment as well, I believe, because then there's a possibility of catching the kidney disease much earlier. I would mention that based upon those studies in Germany, I called someone in the federal government who was responsible for newborn screening recommendations at the federal level. And I did this probably about 2 years ago or so. And I spoke with a very nice lady, the physician, I believe, who informed me that the office was no longer functional. Just the facts. Okay, so any other comments on this? That will
Can I also just say, so I'll put in a plug since we have our industry, and as I'm, partners here, developing newborn screening is complicated, as Dr. Gahl alluded to, in cystinosis. And, in order to really get it in place, partnering with our industry, with folks that are interested in cystinosis, who are developing treatments for it and who have treatments on the market for it, may go a long way towards getting newborn screening into a place where we can consider, starting to advocate for getting it put on the ROS for different states. But that's my opinion as an advocate. And we only have 7 minutes left, and I have a couple other questions. So if you want to wrap up this.
Well, Christy, let me just mention this question here about, because it's related. Can Fanconi syndrome be spotted early via urine screening? In general, the Fanconi syndrome isn't florid or blatant or huge until a little bit later than that. And also urine screening in newborns, it's a little bit tough. I mean, that's why they do the heel stick. That is, it's not so easy to get the urine. Sorry, go ahead. Do you want to ask some more questions, Christy, or address
a couple? And I've had, and they're on different topics. So one, let's get to, since we have only 6 minutes, let's get to this first one quickly, because Michelle asked this a while ago. And her question was, do a lot of kids with cystinosis have dental issues? For example, losing teeth early? I don't know if somebody has a quick answer to that one.
I'm thinking Craig might know a little bit more about that based upon his bone expertise and maybe some of the other nephrologists.
Sure. Thanks, Bill. It's not been, despite the fact that they have florid bone disease, tooth disease has not really been a central feature. So good oral health that you practice would generally suffice, but I'm not aware of any studies that out of the Dental Institute or from my cousin down at the NIH, Dr. Gaffney, that really relate to poor dentition as a major manifestation. Anybody have any other experience different than that?
So I've seen a couple of patients that I suspected they had fluorosis. So they lived in an area where there was lots of fluoride in the water, and they were really polyuric, and they drank tons and tons of water. And so they might have had some dental injury from overdose of fluoride. So I've only seen 1 or 2 like that, but I think it's a possibility because we do see it in nephrogenic DIs as well.
And I would say that I agree with Craig from our experience at the NIH, and we have a lot of our cystinosis patients going to see our dentists.
Okay, let me move to the next question then, and a couple of people have underlined this one. Is there a better method to measure blood cystine levels? Has there been many issues with reliability of the current available granulocyte and leukocyte tests?
So I don't know of a better method. I think that there was an improvement with respect to the preparation of the blood being sent to San Diego. But I think that part of the issue is that there are different methodologies in different laboratories. So, for example, the mass spec method in San Diego is probably, as I mentioned before, the gold standard. whereas some other methods use an amino acid analyzer, which is an anion exchange column, which in my experience doesn't have the same sensitivity. So I think that some of the variability has to do with the actual technological method that's employed in the different places. Jess, you got a hand up?
Yeah, related to that is a low-tech issue that's affected several of my patients. If you go to Quest to have the blood drawn and shipped to San Diego, which I agree is the premier lab, twice I had the tech at the Quest lab, my patient chose say, oh, we don't stick babies. And that PO'd the heck out of me. It was a stormy day and they went there 2 different times. And so I talked to Quest about that, very irate, and they said, well, we don't vet any of our Quest outlets for the ability to stick babies. So that's just a warning. If you're going for the first time, check it out by phone first if you can get a decent answer, because I'm still a little hot under the collar at school here in Michigan today on that issue.
Thank you.
I'd also say that it should be remembered that there is going to be some variability no matter what method you have. And so you should think about treating trends and repeated failed tests. In other words, if it's too high one day, don't necessarily act until you've seen a second one that's also high. Treat trends.
Well, great comment, Paul. And there's a note in the chat that said that your PAM from Verizon can help train the Quest technicians. So that might be something for individuals to pursue.
Yeah, and there's another reminder from Jonathan Dix that Dr. Levchenko is doing a talk tomorrow on a new some work she's doing in developing a new biomarker. So it will be interesting to hear what she's learned so far. So we have 2 minutes left. I'm being asked to wrap up. I want to thank all of our panelists. Bill, I'm going to leave it to you for any final thoughts from the panel, any closing words. And I'll just say before we do that, I look forward to everyone joining tomorrow. You can go in the same way through the attendee hub. And we're looking forward to more research updates and updates from our industry partners, as well as panels that will have parents of individuals with cystinosis and patients or people living with cystinosis on them.
Yeah, I want to thank all of the participants for your incredible questions and the panelists for your answers. And I just like to mention to Paul Grimm that your stockings in the back look like they're going to get on fire. So really be careful of that. And everyone take care.
Everybody have a great evening. Bye-bye.
Bye-bye.
Cheers.
Bye.
Bye.